All right, hey everyone. Thanks for joining us here in person, and to those of you who are joining online. I'm really excited to have you guys here to hear Dalton talk about the project he's been working on. And the reason I'm really excited about it is that it's a great statement of where our NF1 project is at. This NF1 project, in fact, has been ongoing for over a decade. We were just kind of trying to figure out about exactly how long before I even started here at the Biotech Center. And it's amazing to see, how far, this project has come. So it started with some really exciting ideas by Chuck Kaczynski and Danu and Animal Sciences Surgery to come up with an NF1 pig model. And through some, it's kind of amazing what you can do with some real grassroots support through NF Network, through nfteam.org, through Links for Lauren, as well as combining that with some of the amazing cores we have here at the Biotech Center. We've been able to build up this swine genome editing program, and as well as using them to study NF1. So, as we were kind of building up these NF1 tools, and it's been really exciting, and we built this, we generated this data set. We had this data that was sitting there, but we needed the right piece to sort of finish this project off. And luckily, that's when we met Dalton. So Dalton went to college at Cornell, and I guess we would have overlapped there for about a year. So, I mean, he would have been like a cute, chipper, like undergrad, bounding from class to class. And I was like an old, like grouchy, beaten down grad student. But, we passed each other at some point. But he was drawn here to University of Wisconsin because of the storied cancer research program here. He studied progression of prostate cancers in the Wiki lab. But what was great is he built up a huge skill set of all kinds of wet lab skills, from everything from Westerns to cell lines to all those things, to really understand how to tackle cancer research. What was perfect for us was that he was also kind of looking to try something new, and that's about the time we were launching this Biotech Fellowship program. And we were so excited to have Dalton join us to help us understand exactly what these NF1 pig models were telling us. Probably spending way too long talking here. I'll let you have some time. Don't worry. Okay. But I guess I did want to make a quick announcement for the Biotech Talks. This is the last Biotech Talk of the calendar year. There will be more coming up in February, so stay tuned. Check the website for details. I guess with that, Dalton, I'll let you take it away. Thanks.
Okay, thanks for that introduction, Dustin. As he mentioned, this is a culmination of, a decade of work that now we've been able to analyze this single cell sequencing project. And we were able to publish something on that a couple months ago, and that's what I'll be talking about today. So I'll be going through our project of single cell RNA sequencing of neurofibromas, where we found that these tumors reveal a microenvironment that's favorable for neural regeneration and immune suppression, like I said, in an NF1 or Neurofibromatosis Type 1 porcine model. And for those of you who don't know what Neurofibromatosis Type 1 is, or NF1, as I'll refer to it going forward, it's a disease that's manifested over a wide spectrum of symptoms, and it's an autosomal dominant disorder that affects about 1 in 3,000 children per year. Like I said, there's a wide spectrum of manifestations of this disease, as well as known mutations that cause this disease. There's thousands of mutations that have been found in this gene that can lead to symptoms. And so just to go over a couple of those, the most common are these cafe au lait macules, and these are similar to birthmarks that you would get. Typically, you would have 1 or 2 birthmarks. People that have NF1, when they're born, they'll have a lot of these, and usually around the abdomen, they'll have 5 to 7. And that's sometimes used as an indication that you might be affected by NF1. Beyond that, there can be cognitive disorders, as well as cancers such as optic gliomas, breast cancers, leukemias, and skeletal abnormalities. What I'll be talking about today are sort of the main tumor manifestation of this disease are neurofibromas. So 99% of patients with NF1 will get these cutaneous neurofibromas. And these are superficial skin lesions that can be taken off surgically. Some people opt not to, but they're benign and they can be removed. The more severe manifestations start with these plexiform neurofibromas. And these are still benign, but they can get extremely large and they impinge on movement or organs, internal organs and stuff like that. So up to 50% of individuals with NF1 will get these plexiform neurofibromas. And these are, I should mention that these tumors come out of peripheral nerves, or they begin in peripheral nerves. So the architecture of these tumors is extremely difficult when they get to this size to remove, because they're sort of like spider-like, and they invade into other organs and they get really large. So they're extremely hard to remove surgically at this stage. Beyond plexiform neurofibromas are these malignant peripheral nerve sheath tumors. And these are the malignant stage of these tumors. 8 to 13% of individuals with NF1 will get these MPNSTs, as they're called. And these can't be surgically removed or treated. So these are a large burden on patients with NF1. Okay. So like I mentioned, there's a wide spectrum of symptoms, and that makes this disease particularly hard to model. So mouse models, there are a few mouse models for neurofibromas. We set out to create a porcine model. And so this work was about 8 years of work, and it was a huge undertaking to bring this swine genome editing platform to campus. And it involved a lot of cores at the Biotech Center, such as the Advanced Genome Editing Core, Animal Models Core, and then some off-campus sites such as the Swine Research Teaching Center. And so it was a large project to bring this model to campus. So we created 3 different porcine models based on manipulation of the NF1 gene. And so NF1 is a massive gene. It's 350 kb, includes about 60 exons. We created 3 different mutations that would effectively create a altered NF1. The first of which was an early truncation model. So this would, be early on and effectively eliminate NF1. We also created a large excision model. So we took out a 23 kb excision between exons 15 and 30. And then finally, we had this NF1 A31 model. And this was an excision that then created alternative splice variants. And this is also 30% of patients with NF1 will have these varying NF1 splice variants. So it's applicable to humans in that way as well. Okay, so what's unique about pigs, and so this was the A31 model up here. When we harvested the tumors and euthanized this pig, he was almost 4 years old. And the tumors were observed a little after 1 year. So this is unique in that these tumors were able to grow almost 2.5 years before they were removed. So that's atypical of an animal model, especially mice. They don't live that long. So we think that this helped us, and I'll keep touching on that a little bit later, but it certainly helped this tumor to mature and create a mature microenvironment that we could analyze that maybe hadn't been seen before. And so we took 2 tumors on the back of this pig, as well as some normal adjacent tissue as a control. We dissociated those tumors and did single-cell RNA sequencing, as well as kept some tissue for our pathology and immunohistochemistry. And so we had a lot of help in this phase with from the Gene Expression Center and the DNA Sequencing Core, as well as the Bioinformatics Research Center. And I'll just mention sort of some of the growth pathology here. Like I said, these tumors can get very large. It doesn't look very big. These tumors a lot of times don't grow outwards, they'll grow inwards. So, you can see that it's kind of superficial here, but it went pretty deep into the animal and was actually very large. So anyway, we took those tumors and did sequencing on them. But first, the first thing we did was to look at the histology to confirm that we had tumors that looked like, architecturally, they looked like human tumors, as well as molecularly on the single cell side. So our adjacent normal, first we wanted to look at the architecture and the tumor layout. Well, you can see pigs' skin, they have a lot of fat in their skin. You can see that on the adjacent normal, it's pretty much just these pockets of fat filled in with, surrounded by collagen and smooth muscle. And you can see that on the Mason's trichrome stain, which is a stain that stains collagen and smooth muscle. Whereas in our tumors, this area of fat was almost completely filled in. All these adipose cells were filled in and surrounded by tumor cells. And you can see that on the Mason's trichrome as well. These sort of tumor nodules were surrounded by large collagen bands, and then within the tumor, red staining is smooth muscle, so you can see some large vasculature in there. And then one of the things you look for in neurofibromas is heavy collagen deposition, and specifically, when we increase magnification, we're looking at this like wavy collagen. It's called shredded carrot collagen, and that's just really fine wire-like collagen that's deposited within the tumor. And so we can see that here. And that looks about like what a human neurofibroma would. Then we can use some diagnostic stains that are used in the clinic to confirm that it has the same immunohistochemistry that a human tumor would. So two of those stains are the two most commonly used stains are S100 and CD34. S100 is a neural marker or for neural lineage cells. And you can see in our tumor, which this would be staining Schwann cells of the, derived from the peripheral nerve. And you can see we have some nuclear staining there as well as some cytoplasmic staining. And then CD34, which is a marker in for hematopoietic cells of unknown origin in these tumors, but you should have quite a bit of CD34 positivity. And so we saw that as well. And then the absence of that in the normal adjacent. So, we had our tumors looked architecturally like neurofibromas, and they had positivity for our NF1 markers. We also look for sort of differential diagnosis signs. So like these can often be classified as schwannomas, but they'll look a little bit different, and we didn't see anything like that here. We also observed some interesting features in these tumors. So I just wanted to show this to kind of show the complexity and the heterogeneity of these tumors. So we found these zones of hyperproliferative Schwann cells that would sort of form like nodules. And I think the Mason's trichrome really shows how complex this is. You can see how, this zone of proliferating Schwann cells is surrounded by concentric rings of collagen that then it kind of also, you get like a longitudinal outgrowth from there as well. And then you can see that those are staining positive for S100, so they are Schwann cells. And we used a stain for immune infiltrate AIF1. So this just kind of shows that even within these zones that looks to be all Schwann cells, we still had some macrophages that were infiltrating into those zones. And so we spoke with pathologists to nail down what type of tumor this was. In order to call it a plexiform neurofibroma, you have to find the originating peripheral nerve, which we were unable to locate. This is a huge tumor, and this is a really, really small percentage of that. So be hard to find it. We thought early on that this could be that, but, maybe if we had just a couple more millimeters the other way, we might have seen it. But we settled on, or the pathologist recommended that we can call these diffuse plexiform neurofibromas of non-cutaneous origin. So now that we had verified that these were looked like human neurofibromas histologically, now we can start to get into the single cell data. And so one of the first things we wanted to do, or that we needed to do, was to identify all our cell types that should be present in a neurofibroma. So this is our initial UMAP projection up here. And so initially, I think it separated into 14 different clusters, and we were able to merge a couple of those cell types based on markers to create 12 different clusters. And so we used individual specific markers for each cluster. So we identified Schwann cells, which were specified by S100. fibroblasts that were COL1A1, you'll notice that there were 6 clusters that had high expression of COL1A1, and I'll get into that a little bit later. We also identified myeloid cells, neutrophils, smooth muscle, endothelial cells, and T cells. And so we used beyond one marker. So, we had unique markers for each cell type, 3 or 4 or 5, and that's shown here. I think what's most important to take away, besides the fact that we found all of our cell types that should be there, was the cell composition of these tumors. So even the normal adjacent, so each one of these tumors was over 70% fibroblasts, including the normal adjacent, which was 76% fibroblasts. So it makes up a majority of these tumors by far. But beyond that, what you'll notice is we had a couple things jump out. Well, first, I'll point out that you can notice that some of these fibroblast groups are enriched in certain tumors and tissues. And so the red here is fibroblast 2, which I'll talk at length about here soon. And that was enriched in our normal adjacent. And then we had our fibroblast 1 group, which was enriched in the tumors, which I'll also talk about. Each one of these samples had roughly the same amount of cells. So if you're looking for something that should be equally proportioned, it would be, this F3 where you see kind of a third in each one of those samples. Beyond the fibroblasts, a couple things that you might notice, our normal adjacent had higher levels of blue and purple, which are our endothelial cells and smooth muscle, which is what you would expect. And then our tumors had this light tan bar, which is our myeloid cells. So it had an enriched environment of immune infiltrate. And then specifically this dark brown band here, you'll notice isn't present in the normal adjacent. And those are our Schwann cells. So our tumors they were made up of 4.37% Schwann cells. And then our normal adjacent had like 10 to 12 Schwann cells. And these are our, cell of origin for these tumors. So we would expect there shouldn't really be many Schwann cells in a normal piece of tissue, as these are the cells of the peripheral nerve. And so this is particularly important, or this composition of Schwann cells at 4.37% is significant because this is much more similar to the makeup of what a human tumor is. Mouse models overshoot this by quite a bit, anywhere between upwards of 15% of those peripheral nerve tumors in mice can be Schwann cells, whereas in humans it's around 5% in cutaneous neurofibromas. So we're much closer to that composition than what has been achieved in many mouse models. So then once we've identified all our cell types, we can start to look at individual clusters and characterize them and build this microenvironment. So the first group that we looked at were our myeloid cells. And what we noticed was sort of a spectrum of markers across this cluster. So they weren't uniform across this cluster. So we thought that there could be different cell types within here. And so when we did that, SERPA and CSF1R are sort of pan-myeloid markers for porcine specifically. And so this cluster shows that well. But we also identified dendritic cells. And so these are cells that should have high SLA molecules. These are the equivalent of HLA in humans. So you'll see that, the left side of this cluster here has high expression of that. And then they've got low expression of CD163, which is a macrophage marker. So that's what you would see in that group. So we found dendritic cells. We also classified macrophages. These should have high CD206 and MSR1, and then low CD86, which would be an M1 macrophage marker. And so M2 macrophages are polarized macrophages that specialize in immunosuppression and phagocytosis. So this will be important a little bit later. I'll talk about. The unique thing about this cluster was two populations. First of all, we identified this group of fibrotic macrophages. So these were characterized by high expression of collagens. They still expressed, these myeloid markers and macrophage markers, as well as some M1 markers, but they had really high expression of COL1A1, COL1A2, and some other fibroblast markers. So we ended up classifying them as fibrotic macrophages. And these have been identified in pulmonary fibrosis. And so these cells aid in promoting collagen deposition. So we think that, even a small population in this environment would promote increased fibrosis. Lastly, we identified expression of PD-L1 and IDO1. And so these are immune checkpoint markers, and these were expressed in our dendritic cells. And specifically, they were elevated in our tumor cells. So PD-L1 was significantly overexpressed in our tumor groups, in these dendritic cells mainly. We did have one group of dendritic cells that had expression of PD-L1 in our normal tissue. IDO1, on the other hand, was completely absent from our normal cells and was only expressed in the tumor cells. And so this is significant in immunosuppression because it blocks T cell activity, these two molecules. And so overall, we found that the immune cells that were present in this environment were very immunosuppressive, specifically when focusing on the dendritic cells and the M2 macrophages, which made up the majority of this cluster. Okay, so then we can move on to our largest group of cells, which were our fibroblasts. And the first thing we did was separated our tumor fibroblasts from our normal fibroblasts. And you can see that here, how these are distributed. I talked about F2 in our first slide. This was heavily represented by our normal adjacent tissue. And then there were two clusters that were significantly represented by our tumors, and this were F1 and F4. So I'll continue to reference F1, F2, and F4 in all of these later on. So we looked at our normal adjacent enriched cluster, which was F2. And what we saw was that what we would expect. So we saw a lot of normal associated fibroblast markers. And then looking at our F1, we saw a lot of inflammatory cytokines and cancer-associated fibroblast markers that you would expect to see. Specifically, our tumor fibroblasts had high expression of IL-6, CCL2, and other inflammatory cytokines. So this wasn't that groundbreaking, but the next thing that we did was we wanted to identify what each one of these clusters was doing in the microenvironment. So we separated all 6 of these and then compared them amongst each other. So we found so that we could sort of elucidate a role for each one of these 6 clusters in the microenvironment. And so I keep bringing up F2. We looked at, collagens and ECM glycoproteins and regulatory markers and stuff like that. F2 was enriched for most collagens that we looked at in comparison to the other fibroblast clusters. I'll point out a couple that are important. We saw low expression of Col4A2 in that group. We saw low expression of Col5A3 and low expression of Col15A1. And these were all enriched in the other 5 fibroblast groups. And these are particularly important because these are specialized collagens that assist in nerve regeneration. So all 3 of these sort of collaborate to assist in axonal growth, Schwann cell adhesion, and neurite outgrowth. So this is particularly important in this context, and I'll talk about it a little bit more later. Beyond our collagens, we didn't see much difference in the glyco markers, but we did see some interesting regulatory markers that stood out. F5 was enriched for many of these cathepsins that you'll see down here. And these are important because they degrade ECM and make it easier to sort of reconstruct the extracellular matrix in this environment. And so we think that these could be playing a role in ECM turnover and perhaps aiding in laying down these more specialized collagens such as Col4A2 and Col5A3. So the main takeaway being, fibroblast 2 was associated with ECM organization and had high expression of most of our collagens that we looked at. To sort of distill this down a little bit, we took all of our differentially expressed genes. So each one of these groups had hundreds of differentially expressed genes. And then we put it through gene set enrichment analysis to try to nail down a role for each one of these groups. And so this analysis just takes differentially expressed genes and then finds pathways that these genes are associated with and whether or not they're enriched, whether they're overexpressed or underexpressed. And then it can identify pathways that are found in these specific groups. So F1, which was our inflammatory or most cancer-associated fibroblast-like group, was very translationally and metabolically active. So it had really high expression of ribosomal mRNAs. And dysregulation of translation is a common feature of neoplastic cells. So this is something that we would expect. It also had high expression of inflammatory cytokines. Specifically, it had high expression of IL-6 and CSF1. And CSF1 is important in M2 macrophage polarizations. That kind of goes back to the immunosuppression side of things. F2, I won't spend too much time with. Most of those pathways were associated with ECM organization through expression of collagens and regulatory proteins. F3 was that one that was sort of evenly distributed throughout our samples. And it had it was pretty nondescript in its pathways. It had negative regulation of transcription, but it also was enriched for developmental pathways. You'll notice, so these are transcriptional pathways, but then down here we have sort of enrichment of rhythmic process and pattern specification process. So we think that these could kind of be undergoing differentiation or reprogramming by the microenvironment to, one of these other fibroblast types, based on the fact that they have negative regulation of transcription, but also coupled with expression of developmental pathways and differentiation pathways. Next, I'll skip down really quick down to F6 because it was pretty similar to F3. It was nothing really stood out too much from this group. It had a lot of post-translational activity and metabolic activity and RNA splicing. And once again, this is just features that are common to cells undergoing reprogramming or differentiation. The two groups that I'd like to focus on are F4 and F5. Both of these were associated with processes of nerve regeneration, but they were a little bit different. So F4 was our most highly enriched cluster overall in the tumor. So it was made up of 88% tumor cells. And the processes that were enriched there had to do with cell matrix adhesion, cell junction assembly, and stuff like that. And these are processes that are associated with early response to nerve injury. So Schwann cells will upregulate these processes to create synapses and guide axons and stuff like that. So when we looked a little bit closer at some of these genes that are heavily represented in these pathways, we saw molecules that were associated with Schwann cell proliferation in response to injury. And this included molecules like NEGR1 and EPHA3. And these are all associated with response to injury. F5, on the other hand, was more developmental. So these processes were enriched for bone development, skeletal system development, tube development. When we looked a little bit closer at the notable genes within those pathways, we saw SOX9 and Orexin. And so those are sort of context dependent. So tube development within that pathway. There's also neural tube development. So we think that that's probably a little bit closer to what's going on here and associating it with nerve regeneration and development. And we also saw molecules like GRIA2 and LGAUS3, which are associated with gliogenesis. So whereas F4 was more of response to injury, F5 was more developmental and gliogenesis and stuff like that. I'll mention that all these clusters were negative for Schwann cell lineage markers. So we don't think that these were reprogrammed Schwann cells, which is something that can happen, but rather they were resident fibroblasts or recruited fibroblasts that are being reprogrammed by Schwann cells or the microenvironment in general. The last group that we looked at were the Schwann cells, and we saved this group for last, despite the fact that it's probably the most important, because as I mentioned early on, we didn't really have a positive control within our samples because there were none in the normal adjacent. So to do our analysis, we had to compare our Schwann cells to just all the other cell types in here, and to eliminate bias as far as comparing Schwann cells to non-Schwann cells. We used some publicly available data sets. We used a rat sciatic nerve data set, as well as some human sequencing. And what we found was that the markers that were expressed in these Schwann cells were markers associated with dedifferentiated Schwann cells. So we saw markers like CHL1 and Orexin and NGFR and ERBB3. These are all early Schwann cell markers. Most of these markers were associated with Schwann cells that are harvested in embryonic animals, specifically between E13.5 and E17.5. So we think that this is that these are dedifferentiated Schwann cells. And that shows in the gene set enrichment analysis as well, as there were processes of gliogenesis and axon development, neurogenesis. And there were 59 unique genes represented throughout these pathways. So it was pretty heavily represented. And I'll also just touch briefly on IL-34. This was highly expressed in our Schwann cells as well. And this is a potent M2 macrophage polarization molecule. So it seems that the Schwann cells are also assisting in promoting an immunosuppressive environment. So these dedifferentiated Schwann cells are significant because, to wrap all this up, we kind of need to talk a little bit about nerve regeneration. So Schwann cells are unique in that they have the ability to dedifferentiate from a mature state or fully differentiated state and undergo dedifferentiation to aid in nerve regeneration. So what happens is you'll have an axotomy, or a nerve crushing or slice or anything like that. And the first thing that happens is Schwann cells will dedifferentiate. And when that happens, they'll start to release cytokines, and this will recruit macrophages that specialize in phagocytosis to clear debris, myelin debris and axon debris. They'll also secrete some trophic factors and other cytokines that will recruit fibroblasts, specialized fibroblasts that can be reprogrammed to lay down specific collagens to aid in this process. Once the fibroblasts and the debris has been cleared, they can start to form these bridges. And what happens is they form these lines that try to sort of surround the severed axon, and they'll secrete factors like CHL1 and SBP1. And this promotes axon elongation. So they try to guide the axon to the other stump. Once the axon can reach the stump, those Schwann cells will wrap back around the axon and remyelinate and completely differentiate back into a mature myelinating Schwann cell. So it's a unique process in the body, and it hinges upon Schwann cell dedifferentiation, as well as these other important things. So myelin clearance, which is associated with the macrophage recruitment, these formations of bridges and these bands, which are associated with specialized collagens and fibroblasts, as well as secretion of all those cytokines and trophic factors to recruit cells and reprogram cells and everything. So this takes us to our working model at the moment. So what we see is what looks to be very, very similar to a regenerating nerve. And this would be really good for a tumor because it's an immunosuppressive environment and a proliferative environment. So it would be ideal to form a tumor there. Our cancer-associated fibroblasts, I mentioned, we had one group that was expressing these cathepsins, and this would aid in ECM turnover, perhaps to put down these specialized collagens like Col4A2 and Col5A3. They were also secreting nerve damage molecules like FBLN5, and this would help to aid Schwann cells, recruit Schwann cells, perhaps Schwann cell migration. Those cancer-associated fibroblasts were also secreting molecules like CSF1 and IL-10, and this would help to polarize macrophages to M2 macrophages, which specialize in immunosuppression and phagocytosis. Then our Schwann cells were, secreting IL-6, which would recruit fibroblasts, as well as Neurexin and CHL1, which would maybe help to reprogram these fibroblasts and aid in dedifferentiation of other Schwann cells to create this nerve repair environment. They were also secreting IL-10 and IL-34, which would help with M2 macrophage polarization and recruitment. Those M2 macrophages were secreting IL-10, which would promote immunosuppression and tissue remodeling, as well as CCL2, which would recruit dendritic cells. We found that those dendritic cells were expressing IDO1 and PDL1, which would help to block T cell activity, all coming together to create an environment that looks very similar to an environment of nerve repair. Our major conclusions. So our porcine model develops neurofibromas that are similar to human neurofibromas, both histologically and molecularly. Our microenvironment resembles a microenvironment of regenerating peripheral nerves. So we saw Schwann cell dedifferentiation, altered ECM composition, macrophage recruitment with that M2 polarization, fibroblast recruitment, and immunosuppression. And then finally, we saw that our dendritic cells were expressing IDO1 and PD-L1. And this is the first time in a F1 animal model that this has been seen. And I think this all kind of relates back to how long these tumors could grow in our models. So, you know, this tumor had time to grow for over 2 years and really establish a mature microenvironment, and perhaps one that had been transformed from early on. So, you know, from here, what we'd like to do going forward is, can we therapeutically target these critical components of derived from our model, having to do with neurogeneration, to effectively treat or prevent neurofibroma growth? And then also, can we leverage our scRNA-seq data to identify circulating biomarkers that we can detect by liquid biopsy? And this is important because right now, the only way to monitor, or the only way that emergence of these tumors or growth of these tumors is monitored, is through imaging. So, you know, MRI or PET-CT. So that's extremely financially and emotionally burdensome to families. Finding something that we can detect in liquid biopsy would be much more suitable for this. With that, I'd like to specifically thank the NF network for helping us with a lot of this work, and then Dustin, who was my mentor during that time, and Sean, as well as Danu, who was our swine collaborator during this, and then all the cores in Biotech and all the people in Biotech. And with that, I can take any questions.
Right. Thanks, Dalton. That's great. Any of you out there who have questions online, feel free to throw them in chat. Alex, here, let me.
Hey, great talk and great work. I did have a question about the mutation you used in the pig. Is that one that has been done in the mouse, and you got a more translatable result in the pig, or is it something that can't be replicated, you know, homology or some other genetic reason?
Yeah, so it's the equivalent of it in mice is 23A. In pigs, it's 31A. So yeah, I'm not familiar with the mouse model. Perhaps Dustin is, but so maybe he can speak to the mouse model. But it is an equivalent of the mouse mutation.
Thanks.
Yeah, so someone did make a mouse model, but it didn't get the sort of profound panoply of NF1 tumor profiles that we see. So they didn't get spontaneous tumors. They didn't see some of the other things. And they described cognitive defects. And when you have to go through that laborious process of learning and memory stuff, you know, you're searching high and low. Wait, you got to.
So can you talk a little bit about the other NF1 models in swine that are out there, and how yours is similar, better? What's the differences, you know, genetically?
Yeah, I know of one by Nancy Ratner's group. They have a plexiform. They had a female plexiform pig that they sequenced, and it was, yeah, I believe they classified it as plexiform, and it had really high. It was similar to us in Schwann cells, but it had really, really high immune infiltration. So, yeah, that was the difference there. I mean, I can't think of any other major ones. I know that they're studying. They have one for cognitive defects that they're working on, but I don't know too many. In general, there's not.
Yeah, so there are a couple other that have been developed. I think this is probably the most in-depth sort of cellular look, sort of from a genomics perspective. So for the most part,
yeah, there hasn't been. I think that's the only sequencing one, was the one recently by Nancy Ratner's group. That was the only other sequencing one.
Single cell, yeah.
Yeah, but other than that, and then there's only been one human sequencing.
Yeah.
So,
yeah. Yeah, that's right. think I did my stretches this morning.
I guess one question I had is, so you talk about one of the potential avenues of research could be testing different molecules against, how to treat some, these tumors. Your model is better than these mouse models. Do you envision us having to constantly sort of remake or have a stable pig line for that to be developed, or is there would you do some type of cell line creation?
Yeah, we've got it's a great question. So we do have a plan. You can create neurofibroma spheres and inject them into mice sciatic nerves, and this has been done. This has been published. So if you have NF1 het fibroblasts mixed with NF1 null Schwann cells, they create those neurofibromas spheres that then if you inject them into the sciatic nerve, they'll create neurofibromas. So that's a particularly attractive model that we would be able to do pretty quickly. And we want to target, some of these markers that we identified that drive Schwann cell dedifferentiation. If we inhibit them or knock them out, can we prevent this process from ever beginning, essentially?
Thank you. Great work.
Dalton, great talk. Recently, there is a NF1 protein structure that's become available.
Oh.
How does the 31A excision or the 23KB, how does that functionally affect the makeup of the protein and these downstream effects that you see in these pigs?
Yeah, so the 31A is going to create splice variants, and I can't imagine that there's any limit to that as far as the splice variants that it can create. So those would be non-functional, especially if it's, I don't know if they would be non-functional, but and then the excision, I don't know. This is a good question for Dustin. He's the guy who did all that.
No, I mean, that's an interesting paper. Fred's a protein guy, so he can double check. He can either nod and he can shake his head and know that I'm saying something dumb or not. But he might. But as far as I understand, I think that extra exon, is it perfectly situated to either block the RAS domain situationally, I think. And so the idea would be if you're missing that, then you have more constitutive RAS activity. I don't know if that, okay. Brent's saying that I'm not totally full of garbage. There might be something to that. Okay, good. Okay. Cool. Yeah. Looks like we have a couple of questions on chat here. Kyle, thanks. Great talk. As a follow-up to this question, what genetic line of pig was used to create your model? I don't know if that's something.
That is not something that I know.
Sure.
Listen, the Wisconsin pig, right?
No, this we use the conventional sort of production pigs at the time. Starting way back when, before there was a group, genome editing in pigs, we wanted to stick with what we knew. In Wisconsin here, we know conventional pigs, and we have a great resource with Jamie Reichert and SCRT, who is really able to help provide expertise with those sort of conventional like slaughterhouse pigs, right?
Like Duroc or something.
Yeah,
like Duroc.
It's a combination of a few of those strains. Now, alternatively, there are miniature swine pigs, right? In the miniature swine models, and the idea is that those are a little closer in size and smaller. But what we loved at the time was the scalability that we sort of had, the institutional expertise, and we were already kind of ready to take off with those pigs. Another one from Sandy. Thank you. Did you look in the pig brain for these cells? Specifically, EPHA3 and NEGR1 are primarily expressed in the adult brain and promote neurosynaptogenesis and neurite outgrowth.
No, we did not have any of the brain tissue. We looked at some of the brachial plexus. I didn't see anything abnormal. But yeah, I came across that in my reading that these are typically things that are expressed in the brain. So it was very interesting to see them down here, and I believe they can be re-expressed in nerve regeneration by glial cells and stuff like that. So yeah, unfortunately, we didn't look at that in the brain.
Sounds interesting though. Cool. Any last questions here? No? All right. Dalton, thank you for all your hard work on this. Really appreciate it. And thanks for your talk today. Thanks for everyone for attending.
Thank you.